Okay, if you can hear me, we can start now. We were on the physical process models, and we have introduced models for settling resuspension, models for solar radiation, and so on. And today we shall continue with the models that we may employ in order to describe the effect of the biosphere on the light. In particular, we shall introduce the concept of light extinction and look at the model employed for light extinction evaluation. Then we shall go a little bit into more details onto the chemical process of photosynthesis. That is the key process for primary production. So, as I have already anticipated, this will be the core of all your modeling effort during this class. And then, starting from next Thursday, better starting from next week, we will go into the topic algal growth model. So the algal growth model is a model in which we put all together all the concepts that we have introduced up to now. Why we shall discuss about light extinction? You already said that light is the main energy source for ecosystem on the surface of the Earth, and there is a key role in ecological processes like, for example, photosynthesis, transpiration, evapotranspiration, and so on. Hence, the quantification of the energy reaching the Earth's surface and living organisms is important. We have already seen last week an astronomical model that allowed us to evaluate the quantity of solar radiation, so light that is arriving on the surface of the planet, depending on the solar constant and as a function of the photoperiod and of that means the period of a year as a function of the yearly angle of the photoperiod and of the latitude. One moment, because they're asking me. Actually, the slides should already be there under the chapter biological processes, metabolic growth model. No, you're right, algal growth. I have to activate this. Sorry. Okay, now you should have it available on the course web page. Can you find it on the course web page now? The average of the lecture of today?
Yeah.
Okay. So we said that we have introduced last week an astronomical model telling us which quantity of the solar constant, so the solar radiation, reaches the Earth depending on the particular place and depending on the particular period of the year, so depending on the place and time, space and time. But of course, we are completely neglecting what is happening to light once it has reached the surface of the planet. What it is happening to light is a process what we call light extinctions, which means that when light crosses an ecosystem, a water ecosystem, or a forest, or an atmospheric ecosystem as well, as you would prefer, you have a phenomenon that is called light extinction that has to do with the fact that the light intensity lowers. And as you can imagine, as we have always done in this class, we will employ a first-order kinetic linear model describing the effect of light extinction. And in particular, we shall employ the so-called Lambert-Beer or Borger's law, that is the most famous exponential model that describes light extinction. In particular, if we call E the intensity, capital E, the intensity of irradiance, or in watt per square meters, which is the results of our astronomical model that tells us that a certain period of the year and a certain place on the Earth we will have an intensity of irradiance in watt per square meter. This is the output of the astronomical model. Then we may write that the variation in time of the intensity of or irradiance, of intensity of radiation or irradiance, is equal to minus k, the intensity of radiation, times the space crossed by this radiation dx. In hypothesis of an homogeneous media, we can simply rewrite this law as a differential, first order differential law, which we have a derivative in time of irradiance, the derivative, in the space of irradiance, is equal to a decreasing exponential function minus k times E. And as you know, the solution of this equation is this one. The intensity of radiation that reaches an ecosystem of depth z is equal to what arrives on the surface of the ecosystem decreased by this exponential decreasing factor. Of course, this law works quite well for low concentration media, so not too dense media. And of course, all the complexity or the heterogeneity of the model is hidden in the K constants that describes the peculiarity of the model depending, for example, on the direction or on the wavelength. As a matter of fact, if we make some experimental observation, for example, this model that I'm describing here is a water ecosystem. So if we imply with a decreasing exponential model, here in blue, in dot blues, we have experimental measurements. The red line is the exponential decreasing model. We may write that photosynthetically active radiation par is equal to 370 times the exponential of 4.50 times a day. And as you see, the confidence factor of the interpolation is quite high. This law is an exponential law describing the effect of crossing a media the effect on light due to crossing a media. In particular, the Lambert-Beer law is written like this way. In this way, if we write what the transmittance, so the ratio between the energy that is reaching the ecosystem at a certain deep depth, E out, and the energy that is reaching the surface of the ecosystem, E in, then we may write that by application of the Lambert-Beer law, the intensity radiation reaching a depth z is equal to what arrives on the surface, and I recall you that this quantity depends on the astronomical model, reduced, and the astronomical model depends on the time of the year and on the location of the Earth, they're reduced by an exponential factor. Kz is called the optical depth. It is a measure of the ability of the layer to block or to stop radiation or to stop light. Of course, for heterogeneous media, we may apply this relationship by applying different values of K depending on the different condition of the layer that is crossed by the radiation. So, in cases of heterogeneous media, you may include the heterogeneity by simply changing the value of K. There are several applications of this law. Example, the spectrometers. They are trivially applying the Lambert-Beer law of light extinction. And of course, we know that this is due to the fact that not all the radiation approaching the Earth's surface actually reaches the ground. There are several dynamical phenomena. Extinction is due to the two main phenomena that are scattering and absorption. Absorption is due to photons that hit atmospheric gases like oxygen or ozone or nitrogen or gaseous nitrogen or vapor or CO2 or other aerosols, when the energy is transformed into heat directly or irradiated. So absorption is photons that are absorbed by gases. The second light extinction phenomenon is that of scattering. Scattering are photons that are deviated by gases or aerosol without energy loss or diffusion. So absorption is come si dice, una collisione anelastica. Absorption is an anelastic collision, while scattering refers to elastic collisions in which the energy is converted. We can identify two types of scattering: the Rayleigh scattering that applies to particles with a diameter that is smaller than 1/10 of a wavelength. And this is typically applies for gaseous nitrogen or oxygen or for gases in general and for visible radiation. Or we may have what is called Mie scattering, that refers to particles which are much bigger, up to 10 times the wavelength. So particles that interact with wavelength that are up to 10 times their diameters produce a Mie scattering, while particles that interact with the wavelengths that are less than 1/10 of the diameter, they produce Rayleigh scattering. So, how we might describe light extinction in the atmosphere? We apply the Lambert-Beer model. We say that E out, so E as a function of the depth, intensity of radiation as a function of the depth, is equal to E in, so what it is arriving on the surface of a system, reduced by an exponential quantity that depends on some constants. For example, an exponential kml, where m, that is 1 over cosine phi, where phi is the azimuthal, or the height of the sun over the horizon. So you will have a phi of 0 corresponding to a cosine of 1. So you have absolutely no attenuation when the light is arriving vertically. And then to take into account that there are different types of scatterings, we may compose the constant K into different contributions. In particular, the first contribution that is due to the scattering, a second contribution that is due to the scattering by aerosol. So the first one scattering by gases, second one scattering by aerosol, third one is absorbed by gas, and fourth one absorbed by aerosol. Of course, in inhabited regions, usually K of suspended particle is much relevant with K of gases, because in inhabited regions we have a lot of suspended particles that are produced by anthropic activity on the ground. We have empirical relationships for angles that are larger than 60 degrees. That is, I recall you the latitude of circular polar of Arctic circle, and we may also find our value of K by employing empirical relationships and making measurements. So this is the way we describe light extinction in the atmosphere. You may imagine that we may employ a similar model for describing light extinction in water. In this case, we may notice that too few or maybe too much light can limit primary production. So we start introducing the concept of limitation to primary production, limitation to phytoplankton or to algal growth. In the case of water, there may be scattering effects or scattering on absorption effects due to water suspended materials or to water to dissolved substances. But there is also another way around effects, a reverse effect, that is that what we may call the self-shading effect. So that is an effect linked to the fact that primary production itself may limit the quantity of solar energy that crosses the aquatic ecosystem. Into an aquatic ecosystem, there is primary production that is typically made up of microalgae or phytoplankton. This primary production produces a quantity of material in the water column, but has an effect on the ability of light of crossing this water layer. And this is a feedback that we may call self-shading effect. Okay. So how do we model light extinction in water? First of all, we note that the light used in photosynthesis is usually in the visible range, in particular in the range 400 to 700 nanometers. This is the so-called photosynthetic active radiation that may be measured as watt per square meter, so the units of the solar constant, or what may be measured, as we already seen, as the in Einstein, as the photosynthetic photon flux density, so the number of incident photons in the visible range per unit time, per unit area. You may recall the Einstein unit of measure for light. So the Einstein is 1 mole of photons of a particular wavelength. So in particular, photosynthetic photon flux density is the number of incident photons in the visible range. So these photons that are able of producing photosynthetic activity. So when we study a water ecosystem, we may define a neophotic or the photic zone, so a zone in which we have photosynthetic activity, and this zone may be defined as that place of the system which the respiration due to primary producers are PP is equal exactly to the respiration, is equal exactly to the energy produced by photosynthesizer, by photosynthesis, or we may give a more explicit definition by defining that the euphotic zone is the zone at in which we have at least 1% of photosynthetic active radiation. So the zone where photosynthesis takes place at least more than 1% in terms of photosynthetic active radiation. This interval goes from few centimeters to hundred of centimeters to hundred of meters below the surface of the water body. In this case, I recall you, we employ the general model intensity of radiation in water at depth Z is equal to intensity of radiation arriving on the surface, that is the astronomical model, reduced by kz, the exponential of kz, and in this case, k may be made dependent on the wavelength that can be determined using photometers at several depths, for example. And typically we know that the incident radiation at which we are interested is corresponding to the 56% or less, but depends on the wavelength of the radiation that is arriving on the surface of the water body. Why this? Because we know that this quantity of radiation corresponds to the photosynthetic active radiation. 56% is the that part of the solar radiation that may drive photosynthesis. We said that K can be computed in several ways. In particular, we may make K depending on water, K in water and dissolved substances, or on the particulate, or the K of a particulate itself. We have models that link the K of a particulate to the concentration of the particulate. So linear model linking K with the concentration of a particulate. But we must also say that when phytoplankton has the biggest influence, so for example, this is the case of eutrophic lakes, in which we have an important self-shading effect. In this case, you may make K depend on, for example, the concentration of algae in different ways. This is the first linear, and this is the second linear application, non-linear dependence on the K of the concentration of algae. B is in general the background turbidity. In the case in oligotrophic systems, so systems in which we do not have a dystrophic condition, not too many nutrients, normally the background turbidity is what dominates. This way of describing the effect of light extinction is very useful, in particular if you employ various elements for measuring turbidity. You may recall that light extinction in water or turbidity in water may be measured by employing the Secchi disk. Secchi disk is a measure of depth. It is a practical measure in which you make a white disk go underneath the water and you take note of the maximum depth at which you can still see the white disk. In this case, you may employ the Lambert-Beer relationship, the Lambert-Beer model, in which the constant K may be made dependent on various aspects. In particular, we may employ a model that links K to the concentration of algae and to the concentration of the total suspended solids. We have several possible applications of this model. I have just enumerated here 3 possible ways of applying this model. This is a linear application depending on the concentration of algae. This is a non-linear application depending on the concentration of algae. This is a linear application depending only on the total suspended solid. The important point here is that we have a practical mathematical model that may be employed to model explicitly what happens when you employ a Secchi disk measurement, which is the role of a constant k. If we make a plot of a light extinction model according to the Lambert-Beer law, we obtain this kind of curves, saturation curves. In this case, you have two different values of k's. So for larger k's, you have that the saturation is reached more quickly than for smaller keys. This means that the role of k is that of describing the ability of transmittance or the ability of transmitting light at different system depths. Okay, let's go back to the figures that we have seen in the beginning. As I call you, these are empirical measurement made on lake. Here you have the blue points, the blue dots are the data. And the lines are the different model that may be employed for measuring this data. We see that, for example, if we employ a simple regression of the Lambert-Beer model, you obtain the red curve. While if we employ the Secchi disk, so if we make k depending on the Secchi disk depth, we obtain the green model. If we employ the most sophisticated model that we have seen up to now, for example, the dependence of the chlorophyll A and total suspended solids, then you obtain the dotted points. Of course, you see that the dotted points, so this fourth model is the worst one, but also you may recognize that this model is maybe more useful because it is more predictive. So you may, for example, by employing this kind of model, identify which are the processes that most dominate in light extinction. Again, these curves are a plotting of the depth as a function of the photosynthetic active radiations. So by growing with depth, you will have a decreasing quantity of photosynthetic active radiation. Okay. Up to now, we talked about water ecosystem. Now we shall make a small consideration concerning terrestrial ecosystem. How could we model light extinction in terrestrial ecosystem? We start by realizing that the canopy of trees can attenuate light. This is a very macroscopic effect, typically in tropical forest. We may have a significant influence on the underlying vegetation, so the undergrowth, the animals, or the nutrient cycling, or the processes that happens in the topsoil may be strongly influenced by this shading effect due to the canopy. And we have to consider that agriculture, crop space, water balance, and water balance for irrigation are all relevant aspects that may be useful of evaluate. And in order to evaluate this kind of aspects, we need a little bit more sophisticated model, that is a light extinction model in terrestrial ecosystem. To deal with light extinction model in terrestrial ecosystem, we simply make this kind of schematization. We draw a leaf, the leaf of a tree. We say that a portion of the light that is arriving onto the leaf is immediately reflected a portion crosses, so it is transmitted, or what is absorbed. This is the light that is used for performing the photosynthetic process. In order to take into account utilization of light, if you want, in order to take into account the effect of vegetation, we shall introduce a new quantity, but we may call leaf area index, LAI, and we may say that the intensity of light is a function of the leaf area index, so is a function of canopy covering, and again we employ a Lambert-Beer exponential decreasing approach, in which we say that the intensity is equal to the intensity of light that reaches the surface of a canopy reduced by a quantity that is proportional to the leaf area index via the depth Z. So leaf area index Z is the cumulated leaf area index from the top of a canopy to depth Z. So the leaf area index is the ratio of a total one-sided green leaf surface to the surface of the ground underneath the canopy. So it is a percentage, but it describes the quantity of photosynthetic machinery that is available for unit surface in the canopy. K is connected to average leaf orientation. It depends on the tree species. Often, the hypotheses of a Lambert-Beer model are not well suited for this application. In particular, these hypotheses are not realistic in terms of homogeneity and isotropity of the medium. In this case, you may employ other more complex models, like for example 3D vegetation models or field measurements in order to evaluate the effect of light attenuation due to light extinction, due to the characteristics of layers that are crossed by light. Now we shall finish with the lecture by giving a look to the chemical reaction that is responsible of photosynthesis. You may recall that photosynthesis may be chemically written as a certain number of CO2 carbon dioxide molecule interacts with a certain numbers of water molecules, and through utilization of light, H nu, this is a photon of light, it is obtained, it is produced hydrocarbons, C6H12O6, plus what? Oxygen. So this is the prototypical process of photosynthesis. More in detail, we may recognize that there are some limiting factors. Limiting of what? Limiting of the photosynthetic activities. And the limiting factors may be evaluated, for example, by empirical model, like for example, by measuring the photosynthetic rate as a function of the maximum demand for limiting factors. What are the limiting factors? Of course, light is a limiting factor. So we have an optimum irradiance, so the energy is needed, but not too much. Too much energy may stop chlorophyll absorbance from absorbing light energy in the spectrum 4 to 700 nanometers, and hence that may reduce the photosynthetic activity. Our limiting factor is the presence of inorganic carbon. So in order to have photosynthesis, we need some kind of source of inorganic carbon. The presence of water. Water is ubiquitously in all living systems, in particular plants, they need water, so you will have photosynthesis only if there is water available. And then we have the temperature, as happens in all the biochemical reactions, play a relevant role in regulating the speed of reaction. Finally, we may individuate our plant's characteristics or characteristic state of the light, leaf area index, over reproductive states of the availability of nutrients. These are all limiting factors for the production of biochemical molecule, biochemical matter, and the production of oxygen as well. So, if we look then at photosynthesis again, we may say that a quantity of inorganic matter, inorganic carbon, CO2, is interacting with water and with energy. And by this process, we have a production of carbohydrates, so bioorganic molecules, proteins, and so on. We have a production of oxygen. But we discover that not all plants react at the same time, in the same way to light availability. In particular, we distinguish two main behaviors. The first behavior is that of the shade acclimated plants. So these are plants that work well in dark, not too sunny areas. So these plants reaches the saturation level, so a net photosynthesis level, maximum net photosynthesis with a lower quantity of light, while light acclimated organisms or light acclimated photosynthesizers reach the top the asymptotic maximum photosynthetic production with something higher levels of irradiance. Of not only chlorophyll photosynthesis, there are some, for example, autotrophic bacteria with bacteriophile pigments that may make these very strange photosynthesis reactions in which the energy source comes from the sulfuric acid or the electron donor in this case is hydrogen sulfide instead of water, while the carbon source is always CO2. There are also some cyanobacteria that can perform these reactions, so they can change their behavior according to the conditions, surrounding conditions. We call these facultative bacteria. Blue green algae, that is the Cyanophyta most diffused bacteria or cyanobacteria, are unicellular prokaryote, can form colonies. They can reduce nitrogen, for example, fix gaseous nitrogen, or transforming gaseous nitrogen into ammonia nitrogen, this mineral nitrogen, or they can reduce sulfur, or they can reduce chlorophyll, that is typically chlorophyll photosynthesis. I recall you that atmospheric oxygen for cyanobacteria, it is opportunistic condition in cyanobacteria photosynthesis takes place not in specialized organisms, but instead in chloroplasts that are probably organisms that are derived by the symbiosis by cyanobacteria with our symbionts that are able of performing photosynthesis. I recall you also that in chlorophyll photosynthesis, we may individuate two separate reactions, the so-called light-dependent reaction, in which light energy is employed to create high-energy molecules and release oxygen. Remember, is the light-independent reaction in which CO2 is transformed into organic compounds and the energy that can be derived from this molecule may be employed as energy for this process. I recall you that in case of biological processes, we shall describe photosynthesis. We employ the concept of photosynthetic active radiation, but in the end is about 56% of the total incident radiation. And I recall you that photosynthetic active radiation is that radiation with a wavelength in the range 700 to 900 nanometers. In this case, we apply Lambert-Beer application, Lambert-Beer modeling. I0 again is the light intensity on the surface of a water body, and the constant at which you reduce this I0, in this case, trivially, if you deal or you are worried only about photosynthesis, you may reduce directly the I0 that is obtained from the astronomical model by a 56%. So you multiply by 0.56 in order to include the concept of photosynthetic active radiation into your model. More particularly, we may describe more in detail the process of photosynthesis. For example, the typical model employed for describing photosynthesis is this one, the model represented here, in which we say that we have a photosynthetic rate that is equal to a maximum rate that is reduced of quantities. One, on the denominator, we have a ratio between the incident radiation and a quantity of radiation that takes into account the light adaptation capability of the plant at saturation. So EK describes the physiology of the plant. So different plants physiologies, you may have different value of a photosynthetic rate in terms of reduction from a maximum value. But as I also already said, the photosynthetic rate depends dramatically on the temperature of the photosynthetic rate is measured in milligram of oxygen produced per gram of organic matter of CO2 consumed per time. We said that photosynthesis, as all biological processes, has a strong dependence on the temperature. Typically, we employ this kind of equation to describe the dependency on temperature, which is again, you have a maximum value of photosynthesis for a reference temperature of 20 degrees, and this maximum value is reduced by this factor. Or you may also include the effect of pH, because it is also well known that different pH values can influence in a significant way with photosynthetic processes. Like, for example, different pH values may change the ionizing states of various molecules, for example, by tending the equilibrium between CO2 and CO3 minus, moving this equilibrium towards CO3 minus, and in this case, of course, the rate of photosynthesis may change in a significant way. Okay, this more or less ends the biological processes. Now, oh, better next week, sorry, next Thursday, so not tomorrow, but after tomorrow, we will go into more details describing algal growth. So, as I anticipated, algal growth is the last biological process we need to describe in order to build a primary production model for a generalized ecosystem. In particular, algal growth refers to plankton, but grows. Plankton may be zooplankton or phytoplankton. When we talk about algal growth, we talk about phytoplankton. Phytoplankton means plants. Phytoplankton has several very different shapes. These are just pictures taken by electronic microscopy of different plankton cells. You see that the structure may be quite elaborated. This is another picture. Also, this is interesting because you have a reference of the length. Yeah, this is a 5 micron length. This one refers to 5 micron. So this is much larger, 40 micron, 30 micron. So you have a whole set of very heterogeneous shapings of phytoplankton. Well, we know that phytoplankton grows by splitting. This means that each one of these cells at a certain moment, so in particular after the generation time of a particular species has passed, the cell splits into two cells. So from time to time, we have an increasing of the number of algae. Anticipate you the very general model, but is the model on which we shall focus on next lecture, and you shall remember, because this is the typical question to the oral exam, which is the algal growth model or phytoplankton growth model. We write that the variation in time of a concentration of algae is equal, sorry, is equal to a series of factors mu minus r minus exudation minus mortality minus settling. So we have, as you may, as always, the continuity equation, so energy conservation. So we have an additive factor, positive factor that measures the growth rate, mu, and then you have a series of subtractive factors that accounts for the energy consumed by the various processes. You make this model depend again on the concentration of algae. So this is a first order linear up to now linear model. And there is a new element now that we call the grazing that has to do with grazing means brucare in Italian. It has to do with feeding due to hypertrophic levels that feed on the algae. So the G, capital G, represents our connections to the higher trophic levels. Okay, I think I can stop here now. We shall go into algal growth on Thursday, in which we shall discuss limiting factors, and we shall start by temperature limitation. So we will describe how we will introduce the effect of growth limitation due to temperature in our algal growth model. Okay, do you have any questions? Okay, I rest 5 minutes in the classroom. Again, if you have any question, you post questions. Otherwise, we'll see next Thursday.